Hey guys, so I'm going to explain to you a little bit how this kinase is involved with TOR2 signaling. In order for me to do that, I need to explain to you guys a little bit about phosphoinositides. So I just want to make sure that you guys understand that PIs and PTDINS are the same thing as phosphoinositides. So I'm going to be using them interchangeably, and if you see that on the exam, just be aware they're the same thing. You can notice that Marcus calls them as PTD inoticides here, but then here he calls it PI. So just get familiar. For now, ignore all of these words and just focus on this structure. So this is usually how a phosphoinositide looks like. This is a simple PI. And they have these different carbon positions. They're able to be phosphorylated by a kinase at position 3, 4, and 5. They have different functions, and depending on where they're phosphorylated, is what they're going to be doing. As mentioned here, there's different phosphorylated versions of PI, so they're going to function differently at different membranes. You can find these at the plasma membrane, the ER membrane, and the Golgi. Like they're all over the place. He tries to emphasize that they're at different places, like PI3P is at the endosomes, PI4P is at the Golgi. You got this complicated one at the lysosomes. So this is really important for you to understand. And this is like the general concept of what they are. Now, I mentioned kinases. I want to review kinases. Use ATP to add a phosphate group to a protein. So here you see ATP, a kinase comes in, adds a phosphate to whatever carbon position this is. A phosphatase removes it and then takes it back to being dephosphorylated. There are different types of kinases. Not all kinases are called PI. These kinases are specific for these phosphoinositides. This right here, the PI3 kinase, means it will phosphorylate position 3, carbon 3. And that is why you get this image here on the right, where you see a phosphorylation site at position carbon 3. Now, there's another kinase called PI4 kinase that will phosphorylate position 4. This one's very commonly found at the Golgi, and it's known to be involved in Golgi trafficking. That is why Marcus mentioned PI4P is specific for Golgi. Yes, you can find it at the plasma membrane as well. Why? Because there's other kinases like this that make more phosphorylated phosphoinositides. So I was just talking about PI4P. I like to call it HIP. In this, a particular kinase can phosphorylate position 5 as seen here. In order for this to happen, the kinase has to know which phosphoinositide it is going to. That is why you're seeing PTD inositide 4P and then the kinase 5 kinase. So it will recognize this particular phosphoinositide that's already phosphorylated and just phosphorylate position 5. That is what makes it a PI45P2. This 2 is recognizing that there's two phosphorylation sites at position 4 and 5. Same thing goes for HIP3, PI3P, or PI3P, yeah. Um, you have an kinase that will recognize this and then phosphorylate position 5. All of these are involved in a lot of different things. Like I said, PI4P is at the Golgi, but it can also be at the plasma membrane because you need it to make PI 45P2. I like to call this PIP2 and I like to call this PIP. This is commonly found at the plasma membrane, and it is involved in a lot of functions like endocytosis. So let's talk a little bit more about that. How can it do that? How can it be involved in endocytosis? The reason being is because these phosphoinositides have binding domains that can bind to a lot of other proteins. So they have this pH domain that's very commonly known to bind to a lot of proteins that help localize proteins to specific regions and activate signaling cascades. One example is PIP2 in endocytosis. So as I said, you have PI45 PIP2 or PIP2 at the plasma membrane. This was made just from PI4P, okay? And then you had a kinase that recognize the PI4P to make this. So this PIP2 has a pH domain that's able to recruit adapter proteins like this aqua one to initiate endocytosis or the invagination of an endosome to then carry out the MBB pathway so that proteins can be degraded in the lysosomes or vacuole down here. This includes EGFR receptors. It includes transmembrane proteins, right? So it undergoes this pathway. Now, let's look at an example. Let's consider, oh, let's consider that the cell is struggling to survive, okay? And it's determined to survive. What's going to happen is that there's going to be a signal molecule that's going to tell the inactive receptor kinase receptors right here to activate, to be phosphorylated. A good example is EGF. It will bind to the inactive RTKs, which will then allow phosphorylation to occur at the RTKs. So once this happens, it's activated RTK due to the fact that the cell wants to survive, it wants to start growing. This active RTK recruits this PI3 kinase. Remember, it's a kinase. All that this does is phosphorylate things. Don't confuse it with a phosphoinositide. So this is at the plasma membrane still. What's going to happen is that active RTK is going to recruit PI3 kinase, which binds to the RTK, but it also acts as a kinase, right, that phosphorylates our PIP2, our phosphonitrideside 4,5 PIP2 found right here. What it's going to do, it's going to phosphorylate it at position 3, so that's why we end up getting PI 345 P3, 3, that all 3 positions are being phosphorylated. When the plasma membrane notices that this happened, that we have a PIP3, as I like to call it, because all 3 positions were phosphorylated, this causes the recruitment of a protein kinase 1. By it binding to this phosphoinositide PIP3, it is able to phosphorylate something else called AKT. And AKT can then bind to PIP3 or phosphoinositide 345 PIP3. Make sure for your exam you're writing it all out. Don't just say PIP3, okay? Just for the sake of this video, I'm trying to make it simple. So what happens is that this gets phosphorylated, which means it's active and it can then cause a downstream signaling cascade. So all of this is happening via their pH domains, okay? This is important that you understand how kinases work. Do not get confused. The PI3K kinase, this is all it's doing. It's being recruited and bonding to the RTK, and that is phosphorylating our PI45 PIP2 into PIP3, and that itself then causes the recruitment of protein kinase 1 so that it can bind. This together, protein kinase 1 and the PIP3, will go ahead and phosphorylate and activate AKT. So, as I said, it does all of this to survive. It's very important that our RTK receptors work to do this. So, what happens after all of this occurs? Well, you have an activated RTK. Thanks to the help of the PI3 kinase, you activate AKT. That in itself causes a lot of things to happen that you guys really do not need to know. Just understand it activates TOR. TOR signaling essentially prevents proteins from being degraded. It allows for also protein synthesis. It allows for cell growth in general. So, like I said, in the case that the cell wants to survive due to something that's causing it not to survive, a growth factor or a survival signal is the same thing. Okay, an example is EGF will come and cause this to happen. I've explained to you now a couple things. I've explained to you the role of PIP2 in endocytosis, how these kinases come to be. I've explained it more in depth of how it's related to MVP pathway. I've also explained how this particular kinase is able to activate a downstream effect of all of this happening so that you can essentially help the cell survive by allowing more cell growth to happen. overall, if you understand how this is going on and what the role of um, PI3K kinase is, I think you're in a good place. All right. Good luck.